Thank you, Dave, and thanks to the organisers for the opportunity to talk. I'm a biochemist and a plant biologist, so this is a steep learning curve for me these couple of days, and it's a great opportunity. And thanks particularly to Sue for putting on such a great show. So I want to take you into agricultural biotechnology, and in some ways it follows on really nicely from what Adrian was talking about. And I'll try and pick up on some of those themes. So, making crops more resilient to environmental change. I'll touch a little bit on some of our own stuff, but give you a wider picture of what's going on out there in the world in terms of how we're using agricultural biotechnology to meet the challenges of environmental change and all of the other issues around the population growth that we're experiencing at the minute. And the first slide I've put in here is an agricultural technology timeline. In fact, from Matthew Cullen's talk yesterday on archaeology, which we just heard reference to, that interesting point about pottery playing a big part in the beginning of agriculture as well, which I think we should feature in here somewhere as well, which would be good up around the start. But this concept in the context of agriculture, moving from the traditional 10,000 years ago to the conventional late 19th century type plant breeding, which we rely largely on today for the food that we eat, that you ate this morning and yesterday, still a lot of it coming from conventional developments in conventional agriculture back in the 1940s to 1960s. And I'll mention some of this again. Concept of mutagenesis, for example, up to modern agriculture, going through, of course, the discovery of DNA and using molecular breeding, which I want to touch on and give you a context for, up to present day and how we're now using the stuff that we do in research laboratories around the world in terms of improving our knowledge of how plants grow and develop to improve agricultural production. So that's the wider context of agriculture. And then, just to re-emphasize again, this was mentioned in several of the talks yesterday, but just to re-emphasize again this whole concept of the Green Revolution in the 1960s, 1970s that took place, that led to this increase in total agricultural production on a slight increase in the amount of arable land. So this is phenomenal, and this increase, in fact, has kept pace with the growth in human population. It's kept pace with it, or underpinned it, depending on how you want to look at it. And there's a number of processes that have been responsible for this increase in agricultural production: mechanization and irrigation, synthetic fertilizers, crop protection chemicals, and plant breeding and genetics. And this is what I want to focus on. But these are all really, really important, and these are, depending on your perspective, they're good or bad. And quite often, in terms of environmental impact, they're not so good. And so synthetic fertilizers, crop protection chemicals, and one of the things that I want to look at is plant breeding and genetics and how that can be used to address some of these other issues. The other point I want to make on this slide is this all happened during the 1960s, 1970s, and a lot of you will remember the whole development in Europe, especially during the 1980s, where we had these food mountains, and to an extent we still suffer from that whole concept of, a glut of food that we were able to produce in Europe, especially in the 1980s. And we moved away a lot from public good plant breeding and government investment in crop improvement for yield. Yield was almost, a bad word. We didn't need more yield, but we do need more yield. We need to feed the world. And so what's happened is, over the last 20-odd years, we've become more and more dependent on multinational organizations for crop improvement. And the amount of investment that's gone into public good breeding in the developed and the developing world is you know, you could describe it as piecemeal at best. So this is Beddington's summary of it, as we heard reference to yesterday as well, but it does no harm to repeat it. We face a perfect storm of food insecurity. By 2030, we need greater than 50% more food from the same amount of land with less water and more expensive energy and climate change. So that's a tall order, you know, 50% more food. So we need to, increasing crop yields will be difficult and requires major investment. And we need every tool in the toolbox is, you know, if you could one take-home message from my talk, I think that's it. This rather busy and colourful slide is to start to try and cheer us up. You know, plants are fantastic things. They grow, they stay in the same place in the environment, and they've been doing it for 250 million years. And they've evolved a lot to do that. And so they offer a lot to us when we talk about environmental change and how we can use them for the benefit of humankind, for agriculture. And, you know, there's no secrets. It's, you know, most of what plants do are based on 30,000 genes. And if we understand those 30,000 genes and know how they work and know how we can use them to our benefit, then we can use them to our benefit. We can use them, you know, for more sustainable production, less environmental impact. We can use them to improve nutrition. We can improve them for new industries, chemical feedstocks, pharmaceuticals. If any of you know what I'm interested in, I'm really interested in drugs, for good and bad. And, you know, plants are fantastic for all of these things. I'll pick up a little bit on some of the work we're doing, in fact, with Jesse on drought and flooding. But what I want to do is compare now some of the different technologies that we use for crop improvement. And one of the things, again, this was picked up in Matthew Collins' talk on archaeology yesterday morning. The big thing that's happened in you know, across the life sciences and natural sciences in the last decade has been this explosion in our ability to capture DNA sequence. And this is a logarithmic scale of outputs in kilobase pairs, and this is a time frame, and this is when the human genome was sequenced, back in the days when we did Sanger sequencing. And then this happened in 2005, 2006. This is orders of magnitude increase in a single machine run for the output of DNA sequence. So orders of magnitude more for orders of magnitude less cost. So that's meant that you can go in and sequence teeth from people in, you know, 1,000-year-old graves, or you can go out and sequence, you know, weird and wonderful species and discover lots of interesting things about them. So it's a profound impact and will continue to do so. That's why this is the best time to be doing biology as far as I'm concerned. So this is how I talk about agricultural biotechnology and molecular breeding. I'm putting it in the context of conventional breeding that we started off with. And this is natural variation from wild ancestors. I'll return to this in a minute, because this still offers massive amounts of potential for the future, especially now that we've got next generation sequencing and we can discover all of this natural variation in a couple of weeks. Induced variation, which is mutagenesis, this still goes on, went on a lot in the 1950s and 1960s, in fact, to increase the amount of genetic variation that we had to play with for crop improvement. And then this is engineered variation, which is genetic engineering. You know, usually single 1 or 2 or 3 or 4 genes introduced for crop improvement, and we'll look at this as well. And you can go down through, and I've tried to depict here that you follow typically similar processes then of breeding once you've discovered this variation that you're working with, ultimately ending up in new varieties. The details here don't really matter so much. But we now have the tools to follow all of these different types of variation through to getting improved varieties. I just want to dwell for a minute on this comparison now between especially engineered variation and induced variation, because as you all know, this has been a huge topic of public debate in terms of public acceptance for GMOs. So GMOs in the context of plant breeding, so just a few facts. From 1930 to 2007, more than 2,540, quite an accurate number for more than, mutagenic derived plant varieties have been released, 75% of which are crops. So that's I think that's quite a remarkable statistic. Conventional mutation breeding can introduce hundreds to thousands of uncharacterized changes in DNA, many of which remain in the final product. So that's a fact. We can do mutagenesis in the lab. We do mutagenesis in the lab. We can discover an interesting new variety. We can release it next year. That's how it works. And in that time, in the last 80 years, this is a lot of these statements come from this extensive review. We're not aware of any biosafety problems that have been caused. Typically what happens is you go through a rigorous plant breeding and selection process to select the material that's the elite material that's ultimately released. GMOs, in comparison, we have significantly higher hurdles to go over for the introduction of one or two changes in the genome. And so what that has meant, I know, again, directly what that has meant is that to get regulatory approval for a single GMO crop today, you're talking about $100 million at least, okay? So that, because it's like extensive regulatory processes that you have to go through. You have to overexpress any protein that you've introduced. You have to do extensive trials in model systems and lots of other processes as well. So what's happened is for GMOs, the cost has become completely prohibitive. So it's not really possible to apply it to anything other than the equivalent of a blockbuster drug. So you have to have $1 billion product before it pays to take a GMO through to market. So that's essentially where we're at. Public acceptance is dominated by perceived risks in the absence of personal benefits. That's another fact, and so until we have real personal benefits, people are always going to be concerned of perceived risks. So that's. I'm going to come back to GMOs in a few minutes' time, but I just wanted to put that context in so you can think about it and go to sleep now while I talk about conventional breeding for a minute, and then we can come back to GMOs if that's all you're interested in. I put this slide in just to remind us that when we talk about agriculture, again, we're talking about, it's not a natural process. Maize comes from Mexico, it doesn't come from Africa. And, potatoes, as we all know, come from South America, and I'm passionate about potatoes coming from Ireland. So it's, agriculture generally is not a natural process, but we've done fantastic things with it over the last few hundred years in particular. So this is some of the products of conventional breeding. These are tomatoes, these are peppers, all done by conventional breeding, selection. These are potatoes. So you can do a lot with conventional breeding just by selection. And so now we're in the realms of so-called marker-assisted breeding, where, again, using sequencing more than ever before, we can really rapidly discover thousands of molecular markers that allow us to then associate these DNA signals or markers with traits, and we can then select for multiple traits at the same time. And I'm not going to get into the details of it, but this already has had a profound effect on breeding in multinational organizations, but also in relatively small centers like our own. Now we can, because of DNA sequencing being cheap and fast, we can adopt these methods in a whole range of different crop species. And this is just an example from a good friend, Harry Klee, at the University of Florida, who's interested in breeding tomatoes. But he says this: if you're wanting to breed for disease resistance in tomato and also fruit size, then to do that, to stack 3 disease resistance genes by conventional breeding is nigh on impossible at 4.2 times 10 to the 19, 10 to the minus 13 probability of success, whereas if you do it by molecular markers, and you can put out seedlings and screen the DNA, and then take that through, you have a 98% probability of success. So it gives you an impression of what you can now do with molecular breeding. One of the other things which we need to start thinking about more, and this is what we're doing with Sue in the ESSE project that she referred to yesterday, is the whole concept of domestication bottlenecks. And plants have been evolving on land for the last 250 million years. So, and then when we take and domesticate crops, we take them through bottlenecks, and we lose a lot of this really valuable genetic variation. And and in fact, when we look at most of the crops today that we rely on, we're down here, scrambling around in a relatively small gene pool when there's a far bigger gene pool out there that we can be using. And I just put this in. This is, this is just to re-emphasize the whole concept that plants have been on the planet evolving and generating this massive amount of natural variation for all this time, and we're just down here, sort of with a very, very narrow gene pools typically. And so using DNA sequencing and using molecular methods, either GMO methods or molecular breeding methods, we can go back and use a lot of this variation that's evolved with time. That nature usually has a solution out there if we know where to look for it. And this is one example of that rice project which we just started, which is in collaboration with Susan McCouch at Cornell and the Central Rice Research Institute in India. And this is a wild ancestral species or variety of rice, and this is IR64, which is one of the elite cultivars. And the F1s are obviously much better from this photograph. But what's very interesting is you can start to look using those molecular markers. You can chop up the genome of the wild ancestor and systematically put it into the elite variety. This is now a non-GMO method, and very systematically then discover genes which are responsible for things like drought tolerance. But very importantly, the objective of this project is not to get elite high-yielding rice cultivars, but cultivars that produce higher yields under drought. And that's really, really important, because when you hear talk of a 20-ton wheat harvest, it's a 20-ton wheat harvest under completely optimal agricultural conditions. What we're committed to here for the developing world is producing better plants for suboptimal conditions. One of the other points which we need to be aware of is since the beginning of agriculture, humans have cultivated 7,000 plant species, and that's what this is supposed to depict. And then today, only 150 plant species are agriculturally relevant for food and clothing, and 10 plant species provide 95% of the food and feed. So that's obviously an issue in terms of how good are they at producing food and feed under these different environmental conditions. And so again, because the bar is lowered with this revolution in getting into understanding the biology and the DNA sequence behind all of the different species, all of those thousands of different species, we can really realistically, with commitment from scientists and from governments, think about addressing these orphan crops. And we heard reference in Adrian's talk previously about orphan crops or sibling crops. But basically orphan crops are crops that people rely on, but they've undergone very, very little genetic improvement. And we're now at a stage where we can think seriously about improving them. So we have projects running in our centre where you can get significant improvements in some species in 5 years. Is realistic, and get them out into the field in 5 years. There's no reason why we can't be doing more of that. This is an example that's not from ours. It's in fact funded by the Syngenta Foundation for Sustainable Agriculture, but it's in this plant called teff, which is the main cereal that's used in Ethiopia. It's got very low inputs, but it's got very low yield, 0.9 tons per hectare. And this has now been sequenced, or at least partly sequenced, and the genes that are responsible for dwarfing in rice and wheat have been discovered in teff, and people are now systematically modifying those genes by reverse genetics, not GMO, but by tilling to get dwarf teff, which will be a big improvement, since one of the biggest problems with this crop is lodging. This is the main staple cereal in Ethiopia. Right, back to GM. So if you can do all that without GM, why do we need GM or GMOs? So it's fine if the variation that you're interested in is present in that species that you're interested in. But often, and it's not a surprise if life has been evolving on Earth for about 4 billion years, that some of that interesting variation is not in the species that you want, but it's in another species. And that's why GMOs and genetic engineering is so powerful. You can take a gene from anywhere and put it into your favorite crop, in a nutshell. And you don't need sexual reproduction to do that. And so that's the big difference. So this is the global area of biotech crops in going from 1996 to 2011. It's now, 160 million hectares. And in 2011, for the first time, the amount grown in developing world countries is the same as in industrial world countries. So it's about 80 million in each. So that was actually a big milestone in terms of the development of GMO crops. This represents the most rapid uptake in any agricultural technology ever. And, so it's really in Europe where we're still very much behind the wave in the uptake of GMO crops. And this slide, which is very busy, but it basically breaks down the countries where GMOs are grown. There's something now like, I think it's 19 countries growing more than 50,000 hectares. I'll just pick out India, is mainly growing cotton, 10.6 million hectares of cotton grown in India. And I think Adrian also mentioned China. So in China, there's, cotton, papaya, tomato, sweet pepper. The vast majority of it's cotton, 3.9 million hectares. So these countries are adopting it big time. But most of that, as you've seen, are for a small number of traits, these so-called first-generation traits of herbicide resistance, insect resistance. In cotton, it's insect resistance, and consequently decreasing the amount of pesticides that are sown. And now what we see in crops such as corn is stacking of herbicide resistance, foliar insect resistance, and corn rootworm resistance. So you see stacking of, but it's just really focused on 2 or 3 traits. So that massive expansion in agriculture is still largely based on 2 or 3 traits. And part of the reason I was touching on at the start is because of this serious cost in developing GMO in terms of regulatory approval. If we just focus on cotton for a minute, because it's been responsible for this massive expansion in India and China in particular. This is, insect resistant and non-resistant. And I'm not going to get into the details of what's behind it, but there's the publications, the rigorous science behind the safety and the public benefit of this is now well documented. This is from last year, 24% increase in cotton yield per acre through reduced pest damage, and a 50% gain in cotton profit among smallholder farmers in India. And again, in terms of environmental impact, this is a study carried out in China, scaling from 1990 to 2010, over 36 sites, showing 3 types of generalist arthropod, ladybirds, lacewings, and spiders, increasing as an associated decrease in the aphid pests. So, you know, using these transgenics actually is benefiting the environment because we're spraying far less pesticides. So I'm just going to finish with 2 or 3 other examples and then take questions. So just to give you a flavour for what the next 20 years is hopefully going to mean and why it could have a potential impact on production and on the environment. This is one example which comes from this C4 Rice Consortium, which is funded by the Bill and Melinda Gates Foundation. So a lot of you will be aware of the fact that we have different types of photosynthesis, C3 photosynthesis and C4 photosynthesis. C4 photosynthesis is more efficient, and it's evolved to allow plants to capture carbon dioxide more efficiently, let's say. But rice doesn't use C4, it uses C3. So plants like maize use C4. So this consortium is focused on developing C4 rice. It's based at IRRI, and it's fascinating biology because you've got to engineer rice to make bundle sheath cells, basically. You know, if it's I don't know if it's ever going to be a success, but it's at least a grand challenge, and would be exciting if it was. Another example is again funded by the Bill and Melinda Gates Foundation, which was just announced last year. It's led by Giles Oldroyd at the John Innes Centre in Norwich. And this is, a lot of you will know that legumes fix nitrogen, but a lot of other major crops don't. And so what they're trying to do is lift that process that allows legumes to fix nitrogen and put it into other crops. And you know, especially in the developing world. Remember that Africa is where the major increase in population growth is expected over the next 20 years. And so the impact of developments such as this, especially in sub-Saharan Africa, could be profound. I'll finish with this one, which is an old story on tomato puree. I remember going into Sainsbury's when I was a young lecturer in Glasgow, in fact, and buying a few tins of this to show off in a lecture in about 1994, 95. And this was the first GMO product on our supermarket shelves. It was really interesting because, you know, this is a GMO tomato, and this is the wild type. They were both picked when they were fresh, and then the wild type proceeded. This basically was antisensed or blocked in one gene that stops cell walls breaking down. And that's a long, sad story for its demise, partly due to economics, but certainly in Europe due to public acceptance. But, you know, again, food waste and spoilage is one of the biggest issues for food production, especially in the developing world. And technology like this that, you know, allows farmers to get food to market without it spoiling could have a profound effect on profit and on well-being. So this growth is going to continue. So it's up to people like us to, I think, to be critical of it where it's necessary, but to promote it where it's going to be beneficial. And so these are my conclusions. Molecular breeding technologies, marker-assisted and genetic engineering have a massive contribution to make if we're going to feed, you know, a population of the predicted size by 2050 with climate change and with decreased environmental impact. Understanding the molecular basis of complex traits is essential, so we've got to keep on working and doing the basic plant biology work. And it requires foresight and commitment from policymakers, but also from scientists, you know, because we need good fundamental scientists applying their very good brains to some of these big challenges. So, thank you.